Thank you very much. Right, okay, so I've got the task of kicking off after lunch. I notice there was a period of confession from the earlier speakers, all confessing that they might have celiac disease. I don't, at least based on David's point of care testing that I partook in at some point, but that might change, I suspect. Okay, so my task this afternoon is really to give you a flavor of where I think the field is moving, not just in other fields of medicine, but particularly, I think, which is very exciting work that we're results at least looking at celiac disease, looking at the concept of fermentome, and I'll explain a little bit more about that. And I'm sure you're all familiar with the microbiome because that's very topical at this point. Right, so I'll start off with this schematic, which you're all familiar. This is a cartoon cross-section of the intestinal epithelium for the small bowel for all that matters. I think it's important really to understand that this is top lumen going downwards down into the depths of the cell. And you can see here, you've already heard some talks early on this morning with the epithelial cell and then the lamina propria, where your intraepithelial cells, particularly your T lymphocytes, as you can see here, will probably be lounging about. Now, but the focus really, I want to really take you up is into what's going on up here in the lumen, because this is where the food that we're eating, and in particular for celiac disease, the gluten peptides, which are going to be the particular trigger, and how that actually interacts with the microbiome that's there. Don't forget, of course, that there is the mucosal barrier there that has to be contended with, and its subsequent interaction thereafter with the cells. So this is the real area that I'd like you to keep in mind as we move further forward. Okay, so you must be all familiar with this pyramid sort of structure, with the base starting with genomics, which was really the hot topic in the 1950s, moving on then subsequently after 2 decades, 3 decades, looking at the protein and so on and so forth. And likewise now, coming up to the 2020, I like to introduce what I think is the apex of this triangle is volatile markers, because I want to join into the omics frenzy, as I call it, really. And so this is the term which we call and have coined fermentonomics, which is really looking at the cellular interactions and the specific gases, metabolite products that are being produced at cellular level. Still giving you very important information, obviously building on what we know of from genomics and protein data. So, what are these volatile compounds? So basically, they are gases that exist as a gas at room temperature. Now, the important thing about this is that these are fermentable byproducts that we know that occur in the right side of the colon. And as we know that there's much bacteria within the gut, and if these gut are metabolizing these products, they are producing these certain key metabolites which we believe we are measuring. And any alteration, or the term used here is dysbiosis, and we can argue about the accuracy of that term, will result in altered fermentation that we can actually detect. So I've put here a picture of the urine, but you can actually use this principle in any biological fluid, feces, blood, breath, which we're exploring, and even sweat as well. But we're not really interested in what's in the urine, but actually this space here, which is known as the head space. The head is a space above the urine, but just below the lid, really. That's the head space. That's actually key, because this is where the volatile compounds actually lie. And there are different ways of measuring this. You can get an ex-president to do that, as you'll see here, which I think is a rather expensive way of doing things. So we have to look at alternatives in times of austerity. And so how do we actually do this? And the concept actually has come about from very simple, from understanding how we ourselves as mammals smell. So it's really based on the mammalian olfactory system. So when you are presented with a sense of smell, the brain processes this, goes through a set of your neurons, and then you recognize this as human coffee. And this process actually is remarkably intact. We learn it at a very young age. You know, a child at age 4 can actually recognize what a strawberry smells of. The smell of strawberry actually consists of 36 chemicals, but the human nose only needs to smell 4 and it recognizes that this is a strawberry. So this highly defined process still goes on within us as we grow older. And in essence, if you could get a machine to replicate that, then you are quids ahead, really. So what we do is we get some signals, pass it through these chemical sensors. It processes that through a very complex form of artificial neural network, and voila, you get some sort of a probability of what that smell is. And in this case, one of the early results actually was looking into trying to distinguish Brazilian from Costa Rican coffee. And it did that pretty well, actually. But that was almost over a decade ago. We've progressed since then. Naturally, we wanted to look and see how can we progress this to look at human diseases. And one of the early studies that we did going back to 2014 was actually, can we actually detect any key volatiles in the urine of patients with celiac disease? And this was the early pilot data looking at about 36 or so celiac disease patients and compared it to our controls, which were actually those with IBS-related diarrhea. And you can see here on the left, this is a heat map. The heat map uses color to try and tease out any clear separation. And I hope to convince you that on this part of the panel here, you'll see a darker blue sort of coloration depicting those with celiac compared to the more red group, which are the IBS group. And there's clearly a separation there, which I hope you can appreciate even without going into the statistics of that. What was actually more interesting in this pilot group, that when we actually tried to look at identify what are these chemicals, interestingly, for celiac disease, there was only one single chemical that actually stood out, this cyclooctatetraene, which is a complex ester in effect. What we found was that this was present only in those with celiac disease and not with IBS, diarrhea, or even controls. We subsequently filed a patent on this specific chemical because we believe that to hold some promise. So naturally, we wanted to build a little bit more on that and try and work out, well, how can we explore the fermentome a little bit detailed, particularly with celiac disease, because it was proving quite unique in the sense of the chemical that was identified. So we wanted to look at the changes of the VOC signal, but in this instance, what happens when you actually give, when the patients actually go on to gluten? What were the chemicals? Were they any different, or were there some additional chemicals? And then naturally, to show that this was actually probably related to the microbiome, was actually to see there's a shift in the pattern. And then as a consequence of this, and some newer emerging papers since we actually started this project, looking at some very early work on immune-regulated genes and how that links in with the microbiome. So in essence, we took a group of 16 newly diagnosed celiac disease patients, and they had a myriad of symptoms. They all had a raised IgA and IgG tissue transglutamase and had a confirmatory duodenal histology, which ranged from Marsh grade 1 to 3A in effect. We then followed them up for 6 months once they introduced a gluten-free diet. And then at this time point again, we sampled again. And this time we recorded all the patients had symptom resolution. About half anemia had resolved. We also confirmed that they had definite histological resolution. So the villus atrophy had been restored and was healed. So we termed this celiac 1 and celiac 2, and the relevance of that I'll come back to, so keep that in your mind. In terms of controls, we had 3 different controls because this is what we found quite intriguing. The first control we had actually were those with a raised antibody level but had normal histology. Half of them were positive for the DQ2 and DQ8 haplotype. The second control group had both normal antibodies and normal histology. And the third group just had normal antibodies. And this is the group that we followed. So the next step was really, let's have a look at the urine volatile compounds and see if there's any differences in the 2 groups. So what we did here was to see if there's evidence of separation of these patients with celiac disease at the time of newly diagnosed, and then at 6 months later, following introduction of a gluten-free diet. This is what's known as the receiver operating curve. The diagonal line here, which I've pointed here with a lilac arrows, shows a line of no difference. So in effect, if this curve goes closer to this line of no difference, the observation that you see is no different to chance. However, I hope you'll agree that the line here is quite some distance from the line of no difference, giving you a very healthy area under the curve of close to 94%. Now, caution here, the sensitivity and specificity is good, but I've highlighted here the confidence interval is quite wide. But nevertheless, it does show some promise that we can actually detect the difference between a new diagnosis of celiac followed by 6 months introduction of gluten. Now, what happens if you look at the volatiles in terms of separation of the celiac disease patients between the two controls that we talked about? Remember, control 1 were those who had a raised celiac antibodies but normal histology, compared to control 2 who had normal antibody levels and normal histology. And you see the receiver operative characteristic curve is rather similar in that there is quite a large area under the curve, approaching 98% in both circumstances, with good sensitivity and specificity, the caveat being, of course, the confidence interval is wide because of the relatively small numbers. Nevertheless, I hope you are convinced that there is clear separation just purely based on the urine volatile compounds that were detected. So the question you're probably asking in your mind, well, what is it? What is it that we are detecting and seeing the difference? So we decided to explore the chemicals in a little bit more detail. Now, the instrumentation that we use here is a gas chromatography, but it's not your standard mass spectroscopy that's been around for 2 to 3 decades, which is not quite as sensitive. But we used a new sort of machine which is being developed at Warwick called ion mobility spectroscopy. And all that really means is it's looking at the mobility of very small ions at extremely small particulate levels. So it's going down to the parts per billion sort of level. And here you will see here a snapshot. This is what we call a chromatogram. On the x-axis is time, and on the y-axis the abundance of certain chemicals. And you can see here there are various peaks that are occurring. The ones in black are those for celiac disease, and the ones in dark blue dotted about here are the controls. You will see here there are clear peaks that are occurring in those with celiac disease. This is obviously a complete mess in terms of actually what does this all mean. So one would have to drill down a little bit. So we highlight certain areas of interest where we think there's a marked difference compared to controls, and then it populates a table listing of some 211 chemicals that are statistically different. So we've actually teased out all the rest which aren't of interest and narrowed it down to these 211 chemicals that one would have to pour through manually. The reason why it is done manually is because the technique is rather new and we don't have a chemical database as such, but we're building it as we go along. So a different way of actually depicting this is actually the standard box plot curve, which most find it easier to see. Remember, this is looking at principal components. We are not looking at the abundance of differences in the chemicals, but rather a mathematical way of separating the proportion of chemicals that are present in those with celiac disease compared to controls. You will see here in celiac disease that chemicals that are found, the print, if you like, is very unique and very consistent. Very tight, very little confidence interval compared to your controls. So there's a wider spread in actually controls compared to those with celiac disease. In fact, the principle actually holds for most diseases, actually. We see the same sort of pattern even in those with cancer. The chemical print is very tight compared to controls. And the list of, we narrowed it down then to the last 4 of chemicals that actually cause the greatest separation, in addition to the one that we had already shown, which are really complex esters. Now, some of you may already have noticed some familiarity with these two compounds, butanoic acid and acetic acid. These are key metabolites of bacteria that we harbor, mainly the Bacteroides and Firmicutes. So there clearly is some link as to what's going on with the volatile compounds that we're measuring and the microbiome that is underlying these individuals. So the next step was really to try and explore the microbiome a little bit further to see whether we can see if there's a link. Now the technique that we used here is what's called next generation high sequencing. It is rather different to the standard way of doing microbiome analysis where they use a 16S RNA sequencing, where you're actually looking at specific variables only in the bacterial genome. Whereas with metagenomics, this in specifically shotgun metagenomics, we are actually able to sequence the whole sample as it is. You get the entire genome, but also you get classification of not just bacteria, but also viruses, fungi, etc. And you get to see the proportion as a community. So it is a little bit more representative. You get the abundance and diversity, and that's standard. That means you want to know how much there is and how less of one group there is compared to the other. More importantly, of course, is you can explore the functional pathways. What is it actually doing? So to summarize it here on the right here, you do high throughput sequencing. The two questions you want to ask is who is there, and I think we've hopefully going to show you we've answered this bit of the equation. The next question you want to answer is what are they doing? And that's a whole lot of complex pathways to try and understand that. I've selected two of the important ones that have come through because there's a whole masses and masses of data, and I probably need a whole conference actually to actually go through all the data in the microbiome. But focusing on the bacteria and this a phyla group called Firmicutes. You might recall that in the humans, we have four main groups of types of bacteria. You've got your Firmicutes, your Bacteroides, then your ancient sort of Proteobacteria, and then your Actinobacteria. These are the four main groups. We've just picked the Firmicutes because this is the one that had the most interesting and probably the most relevant of which. Now, there were two sort of phyla groups that we looked at, the Blautia species and the Buccoradiales species. We've picked these two because these have never been reported before in patients with celiac disease. So we think it holds great promise and it probably is worth further exploration. But the interesting thing there, if you notice, remember going back to celiac 1 and celiac 2, celiac 1 are your patients at time of diagnosis, so pre-diagnosis, and celiac 2 post-diagnosis 6 months later after introduction of gluten-free diet. And your control 3 were effectively those with a normal histology. You will see here that both these species, actually, the abundance, the relative abundance, still remains low in patients with celiac disease, despite going onto a gluten-free diet. It doesn't really alter. Comparatively to, although you can see here the median is still quite low, comparatively it is still much lower in the celiac disease group compared to the control group. And this holds for this species, these two species. This is when we drill down to the microscope, looking down at the specific species. If we take a step back and have a bird's eye view now, and this is called the Bray-Curtis index, this gives you an idea of the composition index. So this is taking a step back and looking at seeing actually what is going on here, who is actually doing what. The index actually goes from 0 to 1. We've cut the bar short because there's no point, they don't go that far out. If your samples of interest cluster around the 0 mark, all that tells you is that really isn't much difference between any of the groups that you're measuring. So if you see a whole clustering around the 0 mark, it means there's not much difference. The further away you move from 0, you are seeing a difference. And if I can draw you your attention to these star groups, these are your patients who were with celiac disease, newly diagnosed. They seem to cluster around here, compared to the open red circles of those with celiac but on treatment. You can see there's a tendency that they are moving away from what a celiac should look like, but towards what a control should look like. Now, bearing in mind we don't know that, it doesn't necessarily mean that this is what should be normal, but clearly the introduction of gluten 6 months later causes a shift in your microbial communities that you're seeing here towards what we have defined as a control. So this raised a couple of very important questions. Now, we knew that there's some data now suggests that the immune system, but at the intestinal level, this is at the intestinal level, can actually discriminate between those organisms that are pathogenic versus those who are commensal. A very recent BMJ article, which I don't know if any of you seen from the Norwegian group, which was very, very nice, showing an association, mainly with children, of enteroviral infections as babies leading into early childhood as a potential trigger for celiac disease. And finally, this largely ignored paper looking at exosome sequencing, they just did a very pilot data looking at in a whole group of celiac disease patients and looked at exosome sequencings of these rather novel immune-related genes. And they kind of left it out there and nothing really happened. So we decided to pick that up and see, well, actually, is this relevant to our population in the UK? That was a US-based study. And so basically, we replicated based on their 24 candidate genes. We looked at the tissue, look at the exome sequencing in the tissue of our celiac disease patients. We found the 24, but what was interesting was these two, we highlighted only these two genes, TULP1 and MALT1, which not very much is known. TULP1 is tubby-like protein, which has only been described in those with retinitis pigmentosa, and MALT1, which is a murin light transcriptase protein type gene. All we know about them is they are important in immune regulation, in linking in with the bacteria, and we found that they had clear mutations, or C2A mutations, in our celiac disease patients, which we confirmed when you look at expression of the genes, which was clearly much lower, almost fourfold lower than a control in our celiac disease patients. What does this mean? Well, it highlights that there's much work really to understand how these probably novel genes actually regulate what's happening at the bacterial and microbiome level, and therefore the chemicals that we're actually detecting. Because if these regulate the bacteria in such a manner that they produce less or more of certain types of chemicals, that would affect how and what we are actually measuring. So, to conclude, I hope I've convinced you of the utility of urine volatile compounds that can detect changes in patients with celiac disease pre and post treatment, that the chemicals observed do link in quite nicely with the changes in the microbiome that we have observed. Certainly, a lot of these species can't be cultured, and you need to use this high-throughput sequencing to work that out. The shift that we are seeing in patients with celiac disease who have been treated, heading towards what we call the controls. But it leaves for further exploration of these specific genes that we've identified, particularly if the link with the viral triggers has recently been highlighted from the epidemiological study, and also to answer the important question is, what are the metabolic functions? Because we've answered, I think, reasonably well who is there. We need to work out what are they doing. So in summary, we hope to have provided very early insight into some of the cellular changes that are occurring in patients with celiac disease in response to treatment. I just want to finish off and thank, of course, Celiac UK for supporting us in this exploratory work, and of course my collaborators, the engineers at Warwick, Quadram Institute in Norway, who did some of our microbiome analysis, and of course our patients whom we indented to. Thank you very much.
Questions? I wonder if I could ask you a question. Where are you going with this in terms of celiac disease? What do you think the clinical applicability is for it?
Well, one of the things we were trying to explore is actually non-invasive sort of testing. I know there's other markers, particularly to complement blood testing, particularly in children. If you get a urine analysis, which with this technology, it's rapid, and you get an answer within 20 minutes, basically, of rapid, non-invasive diagnosis. The second thing, of course, is using it as a non-invasive marker of shifts of the microbiome pattern in the future.
Thank you. It's incredible work, really a new dimension to understand in celiac. I wonder if you know what's the relationship with gluten consumption and with contamination in the diet. So when patients eat gluten, 90% of the gluten, contaminating gluten in the diet, remains inside the gut. Only 10% is absorbed into the body. So most of that gluten is feeding the microbiome.
Yes.
Is that part of the explanation of why contaminating gluten is so toxic for patients? Is it an indirect effect via the microbiome, and can you use your technology to try to understand that?
Yeah, great question, and that's really what the second part of our study needs to understand is the metabolic pathway. So if we know who is there, We now need to try and understand what are the key metabolic pathways. So are they taking more gluten? Do they sense the peptides and are they responding to that? Or do they preferentially take other short chain fatty acids or other proteins? And I think that will help us answer that. So it's kind of watch the space really.